A biomolecule is any molecule that is present in living organisms, including large macromolecules such as proteins, carbohydrates, lipids, and nucleic acids, as well as small molecules such as primary metabolites, secondary metabolites, and natural products. A more general name for this class of material is biological materials. Biomolecules are usually endogenous but may also be exogenous. For example, pharmaceutical drugs may be natural products or semi-synthetic, or they may be totally synthetic. Types of biomolecules. A diverse range of biomolecules exist, including small molecules, lipids, fatty acids, glycolipids, sterols, glycerolipids, vitamins, hormones, neurotransmitters, metabolites, monomers, oligomers and polymers, nucleosides and nucleotides. Nucleosides are molecules formed by attaching a nucleobase to a ribose or deoxyribose ring. Examples of these include cytidine, uridine, adenosine, guanosine, thymidine and inosine. Nucleosides can be phosphorylated by specific kinases in the cell, producing nucleotides. Both DNA and RNA are polymers, consisting of long, linear molecules assembled by polymerase enzymes from repeating structural units, or monomers, of mononucleotides. DNA uses the deoxynucleotides C, G, A, and T, while RNA uses the ribonucleotides group on the pentose ring C, G, A, and U. Modified bases are fairly common, as found in ribosomal RNA or transfer RNAs, or for discriminating the new from old strands of DNA after replication. Each nucleotide is made of an acyclic nitrogenous base, a pentose, and 1 to 3 phosphate groups. They contain carbon, nitrogen, oxygen, hydrogen, and phosphorus. DNA and RNA structure. DNA structure is dominated by the well-known double helix formed by Watson-Crick base pairing of C with G and A with T. This is known as B-formed DNA, and is overwhelmingly the most favorable and common state of DNA. Its highly specific and stable base pairing is the basis of reliable genetic information storage. DNA can sometimes occur as single strands or as A-form or Z-form helices, and occasionally in more complex 3D structures such as the crossover at holiday junctions during DNA replication. RNA, in contrast, forms large and complex 3D tertiary structures reminiscent of proteins, as well as the loose single strands with locally folded regions that constitute messenger RNA molecules. Those RNA structures contain many stretches of A-form double helix, connected into definite 3D arrangements by single-stranded loops, bulges, and junctions. Examples are tRNA, ribosomes, ribozymes, and riboswitches. These complex structures are facilitated by the fact that RNA backbone has less local flexibility than DNA but a large set of distinct conformations, apparently because of both positive and negative interactions of the extra row on the ribose. Structured RNA molecules can do highly specific binding of other molecules and can themselves be recognized specifically. In addition, they can perform enzymatic catalysis of chiefly fatty acid esters, and are the basic building blocks of biological membranes. Another biological role is energy storage. Most lipids consist of a polar or hydrophilic head and 1 to 3 nonpolar or hydrophobic fatty acid tails, and therefore they are amphiphilic. Fatty acids consist of unbranched chains of carbon atoms that are connected by single bonds alone or by both single and double bonds. The chains are usually 14 to 24 carbon groups long, but it is always an even number. For lipids present in biological membranes, the hydrophilic head is from one of 3 classes. Glycolipids, whose heads contain an oligosaccharide with 1 to 15 saccharide residues. Phospholipids, whose heads contain a positively charged group that is linked to the tail by a negatively charged phosphate group. sterols, whose heads contain a planar steroid ring, for example, cholesterol. Other lipids include prostaglandins and leukotrienes, which are both 20-carbon fatty acyl units synthesized from arachidonic acid. They are also known as fatty acids, amino acids. Amino acids contain both amino and carboxylic acid functional groups. Modified amino acids are sometimes observed in proteins. This is usually the result of enzymatic modification after translation. For example, phosphorylation of serine by kinases and dephosphorylation by phosphatases is an important control mechanism in the cell cycle. Only 2 amino acids other than the standard 20 are known to be incorporated into proteins during translation in certain organisms. Selenocysteine is incorporated into some proteins at a UGA codon, which is normally a stop codon. Pyrrolysine is incorporated into some proteins at a UAG codon. For instance, in some methanogens in enzymes that are used to produce methane. Besides those used in protein synthesis, other biologically important amino acids include carnitine, ornithine, GABA and taurine. Protein structure. The particular series of amino acids that form a protein is known as that protein's primary structure. This sequence is determined by the genetic makeup of the individual. It specifies the order of side chain groups along the linear polypeptide backbone. Proteins have 2 types of well-classified, frequently occurring elements of local structure defined by a particular pattern of hydrogen bonds along the backbone, alpha helix and beta sheet. Their number and arrangement is called the secondary structure of the protein. Alpha helices are regular spirals stabilized by hydrogen bonds between the backbone CO group of one amino acid residue and the backbone NH group of the I+4 residue. The spiral has about 3.6 amino acids per turn, and the amino acid side chains stick out from the cylinder of the helix. Beta-pleated sheets are formed by backbone hydrogen bonds between individual beta strands, each of which is in an extended, or fully stretched out, conformation. The strands may lie parallel or anti-parallel to each other, and the side chain direction alternates above and below the sheet. Hemoglobin contains only helices. Natural silk is formed of beta-pleated sheets, and many enzymes have a pattern of alternating helices and beta strands. The secondary structure or elements are connected by loop, or coil, regions of non-repetitive conformation, which are sometimes quite mobile or disordered but usually adopt a well-defined, stable arrangement. The overall, compact, 3D structure of a protein is termed its tertiary structure or its fold. It is formed as a result of various attractive forces like hydrogen bonding, disulfide bridges, hydrophobic interactions, hydrophilic interactions, van der Waals force, etc. When 2 or more polypeptide chains cluster to form a protein, quaternary structure of protein is formed. Quaternary structure is an attribute of polymeric or heteromeric proteins like hemoglobin, which consists of 2 alpha and 2 beta polypeptide chains. Apoenzymes. An apoenzyme is the protein without any small molecule cofactors, substrates, or inhibitors bound. It is often important as an inactive storage, transport, or secretory form of a protein. This is required, for instance, to protect the secretory cell from the activity of that protein. Apoenzymes becomes active enzymes on addition of a cofactor. Cofactors can be either inorganic or organic compounds. Organic cofactors can be either prosthetic groups, which are tightly bound to an enzyme, or coenzymes, which are released from the enzyme's active site during the reaction. Isoenzymes, or isozymes, are multiple forms of an enzyme, with slightly different protein sequence and closely similar but usually not identical functions. They are either products of different genes, or else different products of alternative splicing. They may either be produced in different organs or cell types to perform the same function, or several isoenzymes may be produced in the same cell type under differential regulation to suit the needs of changing development or environment. LDH has multiple isozymes, while fetal hemoglobin is an example of a developmentally regulated isoform of a non-enzymatic protein. The relative levels of isoenzymes in blood can be used to diagnose problems in the organ of secretion.